and today I'll be presenting on HMGB1 mediated profibrotic gene and ADAM17 expression in neonatal rat cardiac fibroblasts. So first, I'll be going over the disease of interest, which is dilated cardiomyopathy, which is in short for DCM. DCM essentially involves the enlargement and subsequent weakening of the left or sometimes even both of the ventricles. This in turn leads to a reduced systolic function, which in turn leads to a reduced ejection fraction. The prevalence of DCM is that it is the most common type of cardiomyopathy that is found in children, and about 40% of pediatric patients require transplant within 2 years of being diagnosed with DCM. Now I'll be going over myocardial fibrosis that occurs in DCM. Myocardial fibrosis involves the excess collagen buildup that occurs in heart muscle cells. The primary driver of myocardial fibrosis are cardiac fibroblast cells. When cardiac fibroblast cells are under stress or in an injured state, they become activated. Once activated, they differentiate into myofibroblasts. These myofibroblast cells then excessively deposit collagen and other such extracellular matrix proteins. The impact of fibrosis is that it causes the heart to become extremely stiff, and this leads to diastolic dysfunction. And thus, diastolic dysfunction is a much more powerful indicator of DCM compared to other such indicators like ejection fraction. However, there are very limited treatment options for diastolic dysfunction. Now I'll be going over the distinct features present in pediatric DCM compared to adult DCM. In adults, DCM is extremely well characterized. Some of the key features of adult DCM include there's robust cardiomyocyte hypertrophy, there's predominantly interstitial fibrosis, and because it's extremely well characterized, there's a partial response to standard heart failure therapies that exist. But this is in contrast to pediatric DCM, which is distinct from adult DCM, and it is poorly understood. Some of the key differences in pediatric DCM arise as there is no cardiomyocyte hypertrophy observed. In addition, there's predominantly endocardial fibrosis as opposed to interstitial fibrosis in adults. And because pediatric DCM is poorly understood, there's a blunted or even no response to standard heart failure therapies that are mainly geared toward adults in the market as of now. And thus, to study this knowledge and the gap for pediatric DCM, this study aims to investigate the role of secreted factors that are involved in pediatric cardiac fibrosis. So this is some of the preliminary data that was obtained prior to me beginning this experiment. Preliminary data was obtained through a soma scan, protein profile, and we obtained patients with pediatric heart failure, and we grouped them into two. The first group included a non-failing group, which served as a control, and the second group were patients that required transplant upon being diagnosed with DCM. And the protein that was measured was a protein known as high mobility group box 1 protein, which is in short for HMGB1, and their levels in blood serum was measured. And as we could see here in the figure, the patients that required transplant had significantly higher levels of HMGB1 in their blood serum compared to non-failing patients. This indicates that potentially elevated HMGB1 levels could be associated with worser clinical outcomes in pediatric heart disease. And thus we decided to investigate further. So what is HMGB1? HMGB1 is essentially in every resting cell, and it is localized in the nucleus and regulates gene expression. But when the cell is under stress, it becomes passively released, and it becomes a damage-associated molecular pattern. So you may ask, what is its relation to fibrosis? And as we could see here in figure A especially, we see that in the y-axis there's mRNA expression of fibrosis genes, and the x-axis is HMGB1 concentrations. And as we could see, as HMGB1 concentrations increase, fibrosis gene expression increases as well along with it, according to existing literature. This suggests that HMGB1 could potentially induce fibrosis. So we hypothesized that elevated HMGB1 could contribute to pediatric DCM and in general, pediatric heart failure. So we decided to do the experiment where we cultured primary cardiac fibroblasts that were isolated from neonatal rat hearts. Treatments that were imposed, the first being a control, which was left untreated, and then we included a positive control of TGF-beta administered at 10 nanograms per microliter, and then we treated HMGB1 with subsequent concentrations of 10, 50, 100, and 200 nanograms per microliter. Treatments were imposed for 48 hours in total, and the assays used included qPCR and Western blot. So this is the data that was obtained from the qPCR, and we investigated about 3 different fibrosis-related genes. And there's a common pattern with this data, as we could see. As HMGB1 levels are increasing up to a certain intermediate of 50 nanograms per microliter, gene expression is subsequently increasing. But after 50 nanograms per microliter, gene expression is subsequently decreasing. This suggests a nonlinear response where there's a subsequent increase and then a decrease. So what could justify this regulation? And this leads us to another protein, ADAM17, which could potentially regulate HMGB1 levels. ADAM17 is a membrane-associated shedding protein, and it cleaves extracellular domains of proteins. Its relationship to HMGB1 is that it modulates activity of TLR4 and RAGE, which are known HMGB1 receptor proteins. And so now we decided to investigate about ADAM17 and its expression in cardiac fibroblasts. And to do that, we obtained single nuclei ADAM17 expression levels in heart muscle cells. And we saw that ADAM17 expression is exceptionally enriched in cardiac fibroblasts. This was in contrast to other such heart cells like cardiomyocytes. This suggests that there is some prominent role of ADAM17 and its mediated signaling in cardiac fibroblasts. So could ADAM17 and HMGB1 be potentially interlinked? And could HMGB1 induce ADAM17 to increase as well? And that's what we decided to investigate next using a Western blot. And as we could see here in the Western blot data, we did the same type of treatment and same type of conditions, but instead of qPCR and measuring gene expression levels, we measured ADAM17 expression levels. And as we could see here, as HMGB1 concentrations are increasing, there's an increase in gene expression levels, and then up to 50 nanograms per microliter, after which there's a decrease. This is very much similar to the qPCR data that we obtained from gene expression, and this deviates from our hypothesis that elevated HMGB1 could contribute to pediatric DCM. And this suggests that some sort of self-regulation of HMGB1 receptors are potentially occurring in neonatal cardiac fibroblasts. So in conclusion, we see that there's a nonlinear response to increasing levels of HMGB1 in both fibrosis gene expression and ADAM17 protein levels. This intermediate concentration is 50 nanograms per microliter. This nonlinear response could potentially suggest that there's some self-regulation or receptor desensitization in neonatal fibroblasts. So now some future research directions that could be extrapolated from this data is we could evaluate levels of the ectodomains of RAGE and TLR4, which I mentioned earlier as HMGB1 receptors. We can measure them in supernatant and cell lysates. And then we could also compare these patterns to adult fibroblasts to see if this pathway is specific to pediatric cells or whether this could explain regulation in pediatric DCM. So now I'd like to acknowledge the Western Medical Research Conference for giving me the opportunity to present today. I'd also like to thank the Pediatric Cardiovascular Research Lab at UC Entrance, Colorado for mentoring me and giving me the opportunity to work in their lab. I'd like to give a special shout out to my mentor, Dr. Amanda Vincentino, for mentoring me. Thank you.
These papers are open for questions. Go ahead.
So why do you think the gene expression levels decrease after a certain concentration?
Yeah, as I mentioned earlier, I think it's due to some self-regulation, and that's why we came up with Adam 17 as regulating HMGB1 levels, and there may be some like self-regulation patterns that we're not aware of as of now, but that is something that we can investigate further.
So along those lines, you looked at QPCR, but you also looked at protein levels, right? So just remind me, how did the levels down-regulate at the RNA level or at the protein level or both?
So fibrosis gene expression was in the gene level, obviously, and it was there was an increase and decrease, and that pattern was the same in the Western blot with the protein levels as well. So the pattern was pretty much the same for both protein and gene expression.
And what's the biological effect of HMBG1 in general? What is
It's been shown to induce a lot of like fibrosis, and it's also been shown to like implicated in a lot of like inflammatory pathways. Just going to the HMBB1, and when the cell is under stress, as I said earlier, it's passively released, and it becomes a damage-associated molecular pattern. So HMGB1 has been associated with a lot of clinical diseases, and I've looked at research papers and seen that some treatments involve administering HMGB1 inhibitors to treat such diseases. For DCM, there haven't been any like conclusive data about administering HMGB1 inhibitors, but for other such diseases.
So it's a nuclear localized protein
Yes.
that its effects are only when it's been released from a damaged cell.
Yeah, yeah. In a regular cell, it's mostly localized to the nucleus, and I believe it's like unbothered until the cell becomes stressed.
Is it a transcription factor? Does it bind DNA?
Yeah.
What's it doing in the nucleus?
Yeah, it regulates gene expression. I'm not sure if it's a transcription factor or not, but it regulates gene expression. So.
Guess I missed the story. Is Bob back there by any chance? Bob, you know anything about this?
No, but what I was gonna ask is, at the higher levels that you administered in cell culture, did it cause increased cell death?
No, actually, the cells were fine, which was surprising as well. Yeah, they were pretty much like good. I don't know what to say. Yeah, it's just the gene expression was different. So.
And back in Carlin's world, did fibroblasts transition to myofibroblasts very quickly in culture? Did you look at that? I mean, the markers of myofibroblasts like smooth muscle actin?
No. I didn't really look into myofibroblasts, but and like its markers and stuff, but.
Well, that's what you did the alpha smooth muscle actin for.
Well, yeah.
That's the transition to myofibroblast is the expression of alpha smooth muscle actin.
Okay, yeah.
But in your culture, you didn't.
Yeah, we didn't look into like characters.
Yeah. John, you had a question?
Yeah, you also showed some data for TGF beta. Was that upregulated as well, and how does that interact with the HMG?
So the problem with TGF beta, I would have mentioned this. It's just because of timing that I didn't mention it. TGF beta, as we could see here in the results, it really didn't work out as we intended, as their levels were like pretty much lower compared to like HMGB1 at 50. And I actually like talked to my mentors about this, and I think a potential reason for TGF beta being so much lower than expected, because it is supposed to be at a positive control, is because we administered too high of a dose of TGF beta, and that led to it being significantly lower. I'm not sure like the pathways and stuff that are involved in making it low because of the higher dose, but I would assume like some like desensitization. Okay.